So thank you everyone for coming to our next webinar series. We have the great Dr. Rex Bernardo presenting to us. He's the professor and endowed chair in maize breeding and genetics at the University of Minnesota. And before he gets started, I'd like to just advertise a few other things that we have in the pipeline for the NAP graduate students. The next webinar we have in our series is hosted by our commercial plant breeding group, which consists of BASF, Corteva, and Driscoll's, and they will be presenting on the way to my first job. So it's understanding the hiring process and getting students ready for behavioral questions and things like that. So if you're in that part of your graduate career, it'll be a great opportunity to kind of learn what the process is to get hired in some of these companies. And that one will be on April 14th at 8 a.m. Pacific Standard Time. And then on April 13th, we're going to have a coffee hour with Don Cummings. So it'll be a great opportunity to get to learn and talk and network with somebody that's highly regarded in plant breeding, that's very active and helped start the Borlaug Scholars. He used to work for Monsanto, and now he's just very active in ASTA and NAPB, even though he's retired. So it'll be a great opportunity to come learn and network with him. So just a reminder, you can find us on all of the social media. This is brought to you by Renan Souza, who does a fantastic job advertising and getting the word out for all our webinars and other opportunities that we come across. So if you want to interact with us more, please check us out on our social media. So now that I went through that spiel, I'll hand it over to Dr. Rex Bernardo.
Well, hello and good morning or good afternoon or good evening, everybody, depending on where you are. It is a pleasure for me to speak with you this morning, and I so appreciate this opportunity and this invitation from the Graduate Student Working Group of the National Association of Plant Breeders. I think it's a wonderful thing to have these online series of talks and just to be able to hear different perspectives from different people representing different backgrounds. And so today I am pleased to talk about something, that you might not hear too much about. And this is about the great Yogi Berra, great Yankees catcher who had a certain way of putting things into words. And so the title of my talk today is some Yogi Berra-isms and how they are true in plant breeding. I won't be using any slides, so it'll just be me talking with you like this, although I do have some visual aids later on. If you have questions, there'll be a time for Q&A later on. And so I think I might be speaking to you for about 35 minutes or so. And then I hope to have ample time for questions later on. And so again, Yogi Berra had a wonderful way of saying things. One of his quotes that I do like was, he said, "We make too many wrong mistakes." And that makes you think, okay, there's a wrong mistake, of course, it's a mistake, but there's also a right mistake, so to speak, in the sense that, well, it's a mistake, but something right or something good came out of it. Similar way of talking about NASA, talking about a successful failure, that something failed, but it was successful because in the end, there was something good, some success that came out of it. And so, without much further ado, let's look into some Yogi Berra-isms and how they are true in plant breeding. And I think my favorite, if I were to pick one favorite Yogi Berra-ism, is this one here. The future ain't what it used to be. The future ain't what it used to be. And that implies that you and I certainly have some vision of what the future might look like in anything. And here we're talking about plant breeding in particular. And so you and I might have a vision of what future plant breeding might look like. And oftentimes, by default, because there's no other way to think about it, that default is that we think of the future in terms of what we know now. But if you talk to older plant breeders, and I say older like me, and I saw some names in the audience, and I would think they're the same generation as me. And you go back, 15, 20, 30 years back, and you look at the state of knowledge back then, you'd have to say, yeah, the future ain't what it used to be, because what we see today is different from what we thought the future of plant breeding would be when we were at a younger age. So I obtained my PhD in agronomy and plant breeding from the University of Illinois in Urbana-Champaign in 1988. And back then, the focus was just purely phenotypic selection. And so, we talked about, recurrent selection was in vogue back then. At least I was in corn breeding and genetics, and so recurrent selection was a main emphasis. And in fact, my thesis had aspects of recurrent selection to it. And if you were to ask me what I projected about the future of plant breeding back then when I was a grad student, it was phenotypic selection, it was quantitative genetics, it was statistics, maybe a little bit of molecular or biochemical genetics, but for me, that was the future. And we see now, several decades later, that if I compare how plant breeding currently is, it certainly is much different from that vision of a future that I had when I was a young grad student. Never would I have thought of things like artificial intelligence being applied to plant breeding, or drones being able, even existing and being applied to plant breeding, or genome editing being applied to plant breeding. I thought, yes, that molecular markers were coming into the picture when I was towards the end of my PhD studies. And so I had an inkling that molecular markers might begin to influence the way that breeding is done. But again, I go back then and I would have to conclude the future ain't what it used to be, as Yogi Berra famously said. And so we'd have to think of that statement right now and try to see, well, what might the future hold in plant breeding? And there were some questions that were submitted ahead of time before this talk. And a lot of these questions had to do with, what do you think, Rex, what do you think might be future trends in plant breeding, or what's the future hold, or what are some skills I need to focus on, or what are the things I need to be learning now in plant breeding to be positioning myself well for the future. And so when I think of that, I can identify three things that I think would be important for the future of plant breeding. And the first item, or the first field of study or technology, if you would, that I think could be increasingly important in plant breeding is this area of targeted recombination, or modulated recombination, or elevated recombination, or in general being able to exert some control or influence over meiotic recombination in general. If we think of plant breeding and what we do as plant breeders, we have, so plant breeding, as we know, is the genetic improvement of plants for human benefit. So there's a goal in mind, there's a target species in mind, and we want to genetically improve this target species to meet goals that are of benefit to us as humans. And so when we think of that entire breeding process, there are different things that we can control. We can control, for example, what parents we cross. We can control the size of the breeding population. We can control what traits to select for. We can control when we select for certain traits. We can control the extent of phenotyping. We can control experimental designs. We can have a lot of control through that process. And right now, one thing that we cannot control is recombination. We cannot control how alleles at a given locus segregate during meiosis, particularly when there's linkage involved, and we cannot control we have control over fertilization, but it's again a segregation that we cannot control right now. But what if we can exert some control over that recombination process? And there are different ways of thinking about it, and I've been trying to investigate some of these ways. And one way is what I call as targeted recombination. Targeted recombination refers to the ability by a breeder to either select for or to induce recombination exactly or estimated positions where we think recombination would be helpful. And so how that happens is that, well, what we do then is we need to have a way of asking ourselves, where would recombination be most favorable? And for a trait like yield with genome-wide predictions, we can obtain estimates of the effects of markers across the genome. And we can focus at a certain chromosome and say, okay, if recombination happens between marker 1 and marker 2, and we pick the better homologue, what genetic gain do we get? If recombination happens between marker 2 and marker 3, and we pick the better homologue, what genetic gain do we get? And we can walk through a chromosome asking that same question. What gain do we get if recombination happens in this specific interval and we choose that given recombination event? We've found in the studies that students and postdocs and I have done that if we can do that, and I say if because that's a big if we can do that, then we could increase genetic gains dramatically. In fact, we can double the genetic gains compared to selection just based on the random set of recombination events that we have. So again, this, I think, in the future, and I'd like to speculate on how that might look like, there might be a combination of different approaches that allow us to exert some control, perhaps not full control, but at least some control over the recombination process. And for targeted recombination, this could be just relying on natural recombination, meaning selecting for progeny that carry recombination events, ideal recombination events at a given chromosome. For example, line 1 might have a favorable recombination on chromosome 1. Line 2 might have a favorable recombination on chromosome 2, and then we begin to identify copies of these chromosomes from different sources and to pyramid them into a certain line. So in a way, it's just like pyramiding QTL or pyramiding genes or transgenes, except now we're pyramiding recombination events. Now that is a lot easier said than done, because if you think of the number of combinations that are involved, they increase dramatically as you increase the number of recombination events you're targeting. And so we've found, for example, that you can have perhaps target up to 4 or 5 recombination events and have different procedures to efficiently breed them into a certain line. And so that might have to be combined with artificial targeted recombination. For example, when we think of CRISPR technology, we think of it for genome editing, but perhaps for quantitative traits, a more useful way for CRISPR is not actually for genome editing, but for targeted recombination. And there has been recent literature in maize that they have shown that with CRISPR, they can induce targeted recombination that is passed on from parent to offspring. So it's a stable recombination event, just like anything else, except it was induced by CRISPR. And also there have been ways that have been shown to either modulate the rate of recombination across the genome, or, but in my opinion, actually, it's more important to suppress recombination rather than modulate recombination. And it's more than opinion, we've seen this, that because once you have created your targeted recombination, you don't really want to undo that. And so it's important, I think, to have mechanisms to suppress recombination to some extent. And you could see, well, is there something that we could just spray onto the plant? And then when you spray it, the recombination stops, and that, I think, is far-fetched. But if you think about it, there are known environmental factors that suppress recombination. So the number of crossover events has been shown to depend on stress levels, such as heat or drought. So maybe that is that is an active area of investigation. So if I look at a future trend in terms of the future ain't what it used to be, one area that I think of is, again, this whole idea of being able to impart a greater level of control over the recombination process. A second area that I think would become more important is speed breeding, the ability to have as many generations as possible in as little time as possible. Research has shown Over the years, if you look at the Illinois maize experiment in oil for oil and protein, as well as more recent research, has shown that we gain a whole lot more when we have a select and recombine procedure rather than a select only procedure, meaning that we could grow an extremely large population and try to find the best progeny in that population. For example, I have a study coming up in a soon issue of Crop Science that shows that I simulated 10 million recombinant inbreds in each cross, 10 million for a total of more than 1 billion lines simulated. And it was clear that instead of growing 10 million lines in a given cross, it was far better to grow a smaller number of lines, select the best few, then recombine this best few to have the next generation of selection. You get more gain that way than by growing a humongous population of 10 million lines, which means that speed then becomes of the essence, and that we can greatly increase the rate of genetic gain if we can have speed breeding procedures, either by manipulating the growth environment. Are there growth hormones that speed up the process? Are there transgenes that can be introduced and bred out that can dramatically decrease the length of the life cycle of a plant so that we can have this select and recombine process fairly quickly? And you could have trade-offs along the way. You know, for example, I'm working on maize, and for me, it's perfectly fine if instead of having 400 kernels on a maize ear, what if speed breeding allows the production of a corn plant that is extremely small, extremely early maturity, and produces, say, only 20 kernels per ear? To me, that's a perfectly fine trade-off, because I can do a lot with 20 kernels, or even 5 or 10 kernels, and I don't need all 400 kernels to do speed breeding. So that's a second area. A third area that I think would become more important is genetic resources. I think the time has come when, if you look at the different breeding organizations, they're pretty much using the same breeding procedures, at least in corn. They're using using doubled haploids, continuous year-round nurseries. They're using phenomics. Genome-wide prediction is routine. There are differences, of course, in the details of how these procedures are implemented, but what differentiates each program from another is the germplasm base that they have. And I think more attention needs to be given to how do we ensure that there's a steady infusion of new germplasm at the start of the breeding process, so that as we exhaust genetic variation by design, by selecting the best, again, we have new germplasm coming in at the very start and to sustain breeding progress. And so I think attention on germplasm resources and how do we pick what is good from the germplasm while leaving out what we don't want, I think is an area of investigation. One approach that we have used is, instead of looking for QTL in exotic germplasm, we're asking the question, and again, related to targeted recombination, what if we introgress not a QTL, but a certain length of a chromosome segment from exotic germplasm? So we're asking now the question, we're looking haplotype, so to speak. We're designing or creating, asking the question, what haplotype or what block of a chromosome on a given donor would impart the desired changes that we want in our adapted material. So again, if I were to identify three areas: recombination, speed breeding, and genetic resources. Now, you need to notice that these fields did not exist, or we didn't think about it back then. And you may question, and one of the questions said, in plant breeding, we're forced to, there's so much stuff to learn about. And it's difficult to learn about so much material because the field keeps expanding. And so I think one thing that you need to keep in mind is you and I need to be continuous learners. We need to continue learning. But also in grad school, you need to be assured that you have that base of learning that would allow you to pursue different fields according to your interest, meaning that you need to have a solid foundation in genetics, you need to have a solid foundation in statistics, you need to have a solid knowledge of molecular genetics, so that, again, even though these fields do not exist right now, you're well versed, you're well prepared, you're well positioned to pursue these fields in the future because you have that solid footing provided by a solid background in these foundational areas. All right. A second Yogi Berra-ism that I thought is true in plant breeding is this one. You can observe a lot by just watching. Just by watching, you can observe a lot. And I think this is true at least at two levels in plant breeding. One is that you and I need to watch or observe what is going on in allied fields. For example, I think decades ago, animal breeding and plant breeding, there was a big chasm between the two. You had plant breeders on one side, you had animal breeders on the other side. And this chasm was because of fundamental differences in the way that plant and animal breeding was done. For example, you can self-pollinate many plants and you cannot self-pollinate a cow. You can replicate plants and you cannot replicate a cow. On the other hand, it's been said that, well, when a cow is thirsty, it goes to the water. When a plant is thirsty, it stays where it is and it stays thirsty, meaning that genotype by environment interaction is by nature greater in plants than it is by animals. And so plant breeders, there was a generation, my generation when I was a student, when plant breeders strove to have extremely balanced data. We wanted balanced data so that we can do an ANOVA, and the estimation of means was extremely straightforward. In the animal breeding world, no, they did not have balanced data. They had large sets of unbalanced, messy data with lots of factors going on. I think that distinction is much less today, that we see, at least from a data analysis standpoint, plant breeders have become very comfortable with mixed model methodology that the animal breeders pioneered. And so the point I'm making is that you and I need to observe by watching what is going on in closely related fields. So, what is going on in animal breeding? What is going on in robotics? What is going on in the field of machine learning and our artificial intelligence? Because we're seeing, again, plant breeding is the genetic improvement of plants for human benefit, which means that whatever tools that we can use or we can borrow from other fields, we need to be open to them and see how they can be used in the specific work that we're doing. So you and I need to be watchful, which is difficult because there's so much literature already published in plant breeding. And now I'm telling you, well, you need to keep an eye on other fields. But again, that is the way it is. We need to be observant and we need to be watchful. Another way that has become true in plant breeding, you can observe a lot by just watching, is just watching plants and observing plants. And I think there's this notion that maybe even in corn, that a corn breeder does not need to look at plants anymore. And I think there's some truth to that because it's highly quantitative. We're looking at bushels per acre and stock lodging and traits that are quantified and they're very measurable. And yet still, I think there is room to be watchful of the plants, either going through field observation or more recently with the use of drone technology. We can observe a lot just by watching these plants through drones, not just at any one time point, but at multiple time points during the life cycle of the plant. And of course, if you look at other species for which there is aesthetic value, if you think of breeding for roses or chrysanthemums, or even breeding for turf grass or breeding for landscape woody ornamentals, you do observe a lot by watching. And so again, there's where this breeder's eye comes to the fore, and I think it'll continue to be important that breeders need to develop, need to have that watchful eye, and need to be familiar with a particular organism that you are working with, because again, you can observe a lot about the plants, about the subject, just by watching. And finally, the third Yogi Berra-ism that I'd like to talk about this morning, and this is, I think, extremely relevant to postdocs and grad students, is this. If you don't know where you are going, you'll end up someplace else. And I say this in the context of career planning. So you as grad students and postdocs obviously do not want to be grad students and postdocs forever, and you'd like to move on from grad school or your postdoc. And you need to plan your career. You cannot plan everything, but you need to determine where you would like to go in your career as a plant breeder, because if you don't do that, you might end up someplace else from where you wanted to be. Now, if you look at the career of a plant breeder or any professional or scientist for that matter, a career has been categorized into 4 stages. And these 4 stages are as follows. The first stage is when you depend on others. The second stage is when you begin to contribute independently. The third stage is when you contribute through others. And then the fourth stage is when you begin to exert institutional leadership. So first stage, it's when you depend on others. And that's a student, that would be the stage that would characterize you and me when you're grad students. So when you're a graduate student or even a postdoc, you're depending a lot on your advisor, on your supervisor, you're depending a lot on your graduate committee, and it's like an apprenticeship, obviously, and you're learning from those who have come before you. The second stage would be when you graduate or you leave your postdoc and you have your first full-time job, and you need to begin to contribute independently, meaning that as an assistant professor, you have your own, you're on a tenure track, and you have to establish your own research group, you have to recruit your own students, find your own funding, and begin to contribute independently in terms of research outputs that are documented in conference presentations and journal publications, and contribute independently in terms of recruiting grad students and postdocs who you will then mentor. If you're headed towards an industry job, you contribute independently, because now you're assigned an entire breeding program or a part of a breeding program, either largely alone or a part of a bigger team, and independent contributions are expected of you. If you are an enterprising individual and would like to go more for the nonprofit route or a startup route in plant breeding, well, there's a lot, obviously, of independent contribution, because the health and wealth of your enterprise depends on how you do. Now, the third stage then is when you begin to contribute through others. So if you're a faculty member, you begin a lot of your publications now is more of a corresponding author through the publications of your students or your postdocs. Or if you're in a breeding organization, you rise up, so to speak, in leadership of certain teams within that breeding organization. And the last part, which you may or may not be interested in, is whether you want to provide leadership in your organization, either on a formal basis or in an informal basis. Meaning formal, you have a specific title, you are director or lead of a certain effort, or your informal basis in which you basically have your same job title, but you're recognized within your organization as providing thought and directional leadership to the efforts of the group. And so, as graduate students, you need to determine which career path do you want. Do you want to go to industry, academe, a nonprofit? Do you want to exert leadership in an organization eventually or not? Do you want to be more, I want to focus on research and teaching in the long term, and I don't want to ever become a department head or a dean or a center director? These are things you need to be able to determine as a grad student and as a postdoc. Now, that being said, though, your first job does not need to be your last job. And I think that's a main advice I give to the students who come to my group. Your first job doesn't have to be your last job. And your first job is putting your foot into the work world, so to speak, and then charting the path. But again, in your mind, you need to have an idea of where you want to be. Otherwise, you'll end up someplace else. I look at my own career path, and I've been blessed to have different employers over the years. After finishing my grad school, I worked for 9 years with a company called Limagrain Genetics that was located just out in the outskirts of the University of Illinois. Transparently, it wasn't my preferred job when I was finishing grad school, but in hindsight, it was the best job I could ever had. And now after a while, I decided, I really enjoy teaching and working with grad students, and the only way up within a company at that time was to go into management. And at that time, I said, I don't want to go into management or leadership. I wanted to focus on the research that I was doing, and a job opening came up at Purdue University. So I switched careers from the industry to the academe, and then I moved after 3 years, I moved to the University of Minnesota in 2000. And then after about 20 years or so, I figured, you know what, I've been doing this professor work for 2 decades now. How else, when I look at my career progression, am I ready to enter that 4th stage of providing organizational leadership? And that appealed to me, and that's manifested itself recently. I founded and am now director of a plant breeding center that we have at the University of Minnesota. So in a way, it's a different career stage where the expectations and outputs of my colleagues from me are different. I'm doing a lot of my contribution is, for the better good of our organization as a whole, rather than papers or that more papers or more teaching. And so again, it's nice to reflect on a career even though I'm not done. And so my encouragement to the students here is just to think what would be the best fit for you career-wise, and then to go ahead and pursue that path, knowing that your first job doesn't need to be your last job. And there certainly can be changes that you can make along the way. So there you go, 3 Yogi Berra-isms that are true in plant breeding. The future ain't what it used to be. You can observe a lot by just watching. And if you don't know where you're going, then you might end up someplace else. Thank you so much for paying attention to what I had to share this morning. If there are questions, I'd be glad to try to answer them. Thank you.
Thank you, Dr. Bernardo. That was a great talk. And yeah, everyone, if you have any questions, please put them in the Q&A session. We did have one question about targeted recombination when you're not working in biparental populations. I don't know, Rex, if you can see on that Q&A, that question that they had.
Yes, I do. I do. So in case the research does not have biparental populations, can targeted recombination be estimated in bulk segregant analysis? I need to think about this. That would be difficult to do. I mean, when we talk about targeted recombination, we're talking about a highly quantitative trait, and so for a highly quantitative trait, you really could not just separate it, yes and no. I mean, that's a general context, and so my answer would be no. At the same time, I look at targeted recombination not as a academic research, but being routinely used in plant breeding. And in plant breeding, typically breeding is done by crossing parent 1 and parent 2. You could have more than one parent, but typically it's biparental. And so therefore, for the most part, you will have biparental populations that are available. Second question, perhaps related to targeted recombination, would it be possible to make use of meiotic drive mechanisms to bias recombinants that you want against those you don't want? I don't know. I don't know the answer to this question. I do not know enough about meiotic drive mechanisms. What I do know is that in rice, as well as in Arabidopsis, there have been genes that have been shown to elevate or to increase the rate of recombination genome-wide. So again, it's overall increasing the recombination rate. The thing is, typically, obviously, when you have a recombination, one you want, the other you don't want. And if the question is, are there mechanisms to increase what you the homologue that you want? I think that would be less of an less important than just being able to target it where you want.
Thank you, Rex. We can maybe go to some of the questions that we had submitted, or that people had already submitted. Here's an interesting one. What are 3 skills you think a plant breeder can't live without?
I think that's a great question. I saw so there were some questions that were submitted in advance, and what are 3 skills a plant breeder can live without? And obviously, yeah, you need many skills, and you need, and if you don't have those skills, you need to be working with people who have those skills. But if I were asked to choose 3 skills, here's what I came up with. The first skill is what I would call as numeracy. We've heard of the term literacy, the ability to read and write. And for me, numeracy is the ability to interpret, to analyze, to make sense of numbers. In plant breeding, especially for quantitative traits, we deal with numbers all the time. So a certain comfort level with numbers, either inherent or natural or acquired, I think is extremely important. And again, I call that as numeracy. A second skill that I think is important is communication. Plant breeders, the era of breeders working by themselves in isolation is long gone. When I started my career, in a way, that was the model. You had one breeder at one research station with 3 technicians, 1 secretary, a bunch of tractors and planters, and a bunch of combines. Now, breeders work in a team. And the ability to communicate well in a team cannot be overemphasized. And that communication is not just with peers, but even with those who work under your supervision and those who work above you, those who supervise you. You know, for example, it might not be uncommon that you would have a CEO or somebody higher up who might not be a plant breeder, but might have a background in, say, plant nutrition or nutrition, human nutrition. And if that were the case, well, how do I communicate well with this other person, knowing what my message should be? What does this other person need to know from me, and how do I communicate that well? So numeracy, communication, and the third is general playing well in the sandbox. I think there are a lot of interpersonal skills that are necessary because, again, of breeders working in teams and playing well with others in the sandbox is extremely important. If I may mention a 4th skill, I think it's project management. A lot of the breeding work these days is delegated, right? So double haploids are made by a team here, and marker genotyping is done by a team there. So there's a lot of project management that has to be done so that you can have your deliverables, which in the end are cultivars, delivered in a timely fashion and in an effective manner. So those are the 4 skills that come to mind.
We actually had another one in the chat. What has been key for you to adapt all the changes you had to make in your career? And then
what's the key? Say again, what's the key?
Yeah, it says, what has been key for you to be able to adapt all the changes you have made in your career?
All right, it's a great question. One is to know that change will occur, whether you want it or not, change will occur. And the second is to be a learner, to have this mindset of I want to learn on a continuous basis so that I'm not caught behind or I'm not left out, so to speak, when these changes occur around me. My advisor had this quote, and I'm going to paraphrase it. It was attributed to a man named Eric Hoffer. in his office, and this quote stuck with me, and it's like this: in times of change, learners inherit the earth, but the learned find themselves beautifully equipped to deal with a world that no longer exists. Meaning that if I'm learned, that I know a lot and I'm confident in what I've learned, but I'm not willing to learn, then I might find myself basically as a dinosaur. I can function well in 2040, but this 2040 world, or this 20, I can function well in 2020, but this 2020 world no longer exists in 2040. So again, it's a fairly broad answer to that question, but I think, again, it's that mindset of, you know what, I need to be continuously learning. And do I do that well? I would say in some aspects, yes. In other aspects, no. For example, I grew up in the era of SAS for statistical computing. And now R is the lingua franca in statistical computing. SAS I can still use, obviously. And I had this ambition, I'm going to learn R, and I downloaded the manual, and I looked at the manual. I said, no, I'm not going to do that. It's just too much hassle for me at this time when I can have a fallback with another software. What do you think about subpopulations as a fixed variable? You can have it as a default. You can fit subpopulations as a fixed variable in genomic predictions, and you can have it as a default, meaning that if it is useful, then it would have a contribution, and if it's not useful, then it's just going there for the ride, and it's not contributing anything, but it's not hurting you either, right? Meaning that when you say subpopulations, how they're typically structured is you have a subpopulation effect, meaning that if I have flint corn and dent corn as my two subpopulations, it could be that kernel hardness would be, and I'm looking at kernel hardness, then the effect would be I have a subpopulation effect for kernel hardness attributed to flint corn, harder kernels, dent corn, softer kernels. And so in that case, it would have an influence. But if I have a subpopulation effect, flint corn versus dent corn for something, let's just say plant height, if flints and dents are of equal height, then that subpopulation would not have an influence. It's just there, the subpopulation effects were zero. It won't help, but perhaps it won't hurt either. So the default is, yes, if you think it would be important, then just include it as a default. It's a great question. If someone does not want to follow the normal career path and remain that contribute independently all their career, is there not a risk they eventually become obsolete due to the fact that it is hard to continuously train and work at the same time? Then it is not almost inevitable to eventually work through other and end up in leadership positions. Indeed, what has been shown is if you spend a long time in a given stage of your career, the performance evaluations decline. And there's literature that shows that those who have stayed too long at a given stage, their performance evaluations decline for this exact reason. And so it's inevitable that in a way you work through, if you want to be successful, that you work through this natural stage. Now, you may not end up in the last position of providing organizational leadership, just because if you look at, say, a 30, 35, 40-year career, then there's enough of the other stages to fill that entire career.
There's one from the the submitted questions before that I think might be relevant to kind of what you talked about. You mentioned AI and things like that. How close do you think it is for like deep learning and AI to replace some of the other older prediction and technologies in plant breeding?
Yeah. So this is just a guess. This is my opinion. I don't think it will replace. I mean, so it can do some things, right? But as far as replace, no. Perhaps it can do something adjunct to what's being done, but replace not necessarily. The thing with AI is you need extremely large data sets, and those data sets certainly would be available in major companies, but in smaller organizations, even university breeding efforts, the size and the scope of the data set might not be available for them to make an impact. Now, if you look at genome-wide prediction, so far the literature has said, it may help a little bit, but really not that much. That if you look at it, ridge regression blob and G-blob, for the most part, remain solid procedures for genome-wide prediction. If you use AI, it might increase predictive ability by 0.02, 0.03 here and there, but typically not by a large increase. There have been efforts by companies to try to predict the selections that a breeder would eventually predict, and I think that's an important field. Whether or not you need AI, for me, is a question, because there are some classical procedures, something that's called as retrospective indices, developed in the 1950s. Retrospective indices help you or allow you to estimate the weights that are intuitively used by breeders for each trait. So perhaps I think there's a limited literature that's shown that I published back in the 90s, retrospective indices help predict what breeders would select. And so whether AI would be useful, I'm not quite sure about that. But certainly it can be used as an adjunct if AI can be used to, delete or eliminate, say, 60%, 70% of the candidates. That would allow the breeder to then focus more closely on the remaining candidates that might be predicted to be the best.
So kind of focusing on that, what do you see as the next trend topic in plant breeding, and what do you think would be considered old but still working?
Selection works. Selection always will work. When I think of trend topic, I think I mentioned that the targeted recombination, and I think really being able to do many generations of selection per year would be an important topic. And the question in the chat: do I safely assume speed breeding you described would be the same as being used in rice and other crops to 2 to 3 generations within 1 year? And when I say speed breeding, I'd like it to be more faster than that. What if we can grow a crop in a month? Set our sights high. What if we can go from seed to seed very aggressively in a month? And how you do that? Well, maybe you need to do things like to combine it with other things, such as old things like embryo rescue, right? If I rescue the embryos, then I skip that seed maturation and germination process. So that buys me a lot of time. But it's not just that. What if I manipulate day length, heat, water, fertility, plant hormones? How can I have seed to seed from a month? We can do it in Arabidopsis, I guess, or the fast plants. Can we do the same sort of thing in other crops? And if so, but then how do we do that? So in a way, it's like this moonshot thing, right? But yeah, if we really could speed up the generation interval, then we can really speed up breeding progress.
Do you have any of those questions in the Q&A that you would like to answer? There's 3 more, I think, that we haven't.
Sure. If I want to work as a private plant breeder in non-private institutions, what places should I consider? Well, right now they are universities, the USDA. There are organizations such as the Land Institute, for example, more of a foundation, or the Noble Foundation out in the Southwest that you could look into. And so I think in a way, industry is sort of monolithic. They have characteristics. Then you have the academe, and then you have the non-private. That they are a lot more diverse. So it's more a matter of knowing what's out there and picking what you think is a best fit for you. World Food Organizations like the FAO, I'm not familiar enough with organizations such as the FAO. I would need to say, not picking on FAO, but just international research institutes in general, a continuing problem is funding. Oftentimes, you sign a contract to be a breeder there for 3 years at a time, so funding is an issue. The lack of funding just makes it difficult to have longer-term breeding programs that we need for the crops that we work with. And also, just in general, not as a bit of a criticism, but more realities. Many of the organizations, such as what I've mentioned, change happens slowly. Change happens very slowly, and I see that as a bit of deterrent of clinging to how things have been done, and perhaps not willing to embrace just the new developments that are coming up. The most useful tool that plant breeders can adopt from animal breeding right now, I think, is, again, just this whole mindset. And I think we do have it. It's just mindset of thinking of big unbalanced data. Again, if I look 3 decades ago, 2 decades ago, we as plant breeders weren't there. But now we more and more as plant breeders, we're thinking like animal breeders of having big unbalanced data. And there where we come up with genetic, for example, where we update the genetic evaluations from year to year. For example, in animal breeders, they could have records of, these are the breeding values for the dairy bulls. And as new records come in, then these records become updated. And so I think that mindset is something we need to adopt more in plant breeding. That let's say we have a pool of 200 lines, elite lines, and then so we should have yearly we have estimates or predictions of the value of each line, and that we have that in a book or some spreadsheet or something. And then as new data become available year in and year out, and then these get updated. I don't think we're that systematic in that, these are the latest and greatest estimates of the net merit of the lines we're working with for different traits. Question here, how do you see the potential for renegades who want to start their own breeding company? It's a great question. One is make sure you have money to buy food and pay for rent or pay for your house. I mean, when you just want to start your own breeding company, it certainly would entail a lot of risk, and you want to be careful as you do that. So in a way, you need to have some fallback option at the same time. If you want to start your breeding company, it would require being all in. It would require a certain mindset. It would require a certain way you're put together, so to speak. Not everybody can do that. I've actually thought in the past of starting a breeding company, and my non-novel idea was just going back to ex-PVP lines that I had. So when I was with the industry, we were working with lines that are now with Monsanto, and I knew the best combinations of these lines that would make hybrids. Well, now they're being off PVP, and I go, oh, I can get the seeds from publicly available, and I can make those same crosses. Will they be competitive with the hybrids today? Not at all, but I could sell them very cheaply, the seeds very cheaply, for a half or a third of the price of a bag of corn seed that way, and make money that way. Well, that just remained an idea, but that never came to fruition. But Josh, if you want to partner with me in that effort, maybe we should talk or something like that.
There was one question that I was pretty interested in from the submitted questions is, so on the future of the plant breeder themselves, what is your thoughts on, as plant breeders become more data driven, if traditional plant breeders actually even needed?
That's a great question. I've thought about that too. You know, if you can automate, the entire process, how many plant breeders would still be needed. But I think the question is, how many do you need? How many do you need? And I would have to say that we need fewer than today. And so let me focus more on corn and soybean breeding, because these are more the species with a lot of effort from the private sector. Yeah, a lot of those processes can be automated. I mean, you could have machine learning algorithms or quantitative genetics approaches that could suggest, you know what, these could potentially be the most successful crosses, breeding populations that you can make. Okay, among the progeny that are being developed in the breeding program based on the data, you know, these are the data. I mean, these are the progeny that are most needed. And so, you know, it then boils down to the question, what's a breeder left to do? And, you know, I think there's always room for creativity. I think one area where a breeder's input will be helpful is just responding to the unexpected. There was a famous rocket scientist, German, immigrated here, Werner von Braun, and he said, you know what, talking about the space program, he said that one thing humans can do well is anticipate or react to what is unexpected. So you have a machine learning algorithm, and it encounters something unexpected, it might not know how to react. Whereas if you're a trained, educated plant breeder, you have something new, you can fall back in your knowledge, your experience, your background, your intuition, and say, you know, this is how to address this issue that just came up. But yeah, it's a great question, and we'll see. And I think it's an important question because when you look at publications in plant breeding over the years, a main thought has been, and I think it still is true, at least today, that we need more plant breeders. We need more plant breeders. We need more funding for plant breeders, training for plant breeders. And we need to ask, if we're serious, how true is that? Will that continue to be true in the future, or will there actually be a lower demand for plant breeders? And if so, then, and if there's a lower demand but a new skill set, how many do we need, and what skill sets do the new plant breeders need in a decade or 2 or 3 decades?
So maybe one last question, kind of building off of that. There's a question in the chat that says, there's been a big concern on the training of future plant breeders. What can be done at universities and private industry to ensure a better future for plant breeding?
It's a great question. I mentioned the skills that are needed, and 2 of those skills are actually non-technical skills, non-science skills. So I think a lot more emphasis has to be placed onto training and professional skills. I teach a professional skills course for scientists here at the University of Minnesota, and I'm hoping that such a course will become more prevalent across different universities. I think it calls for the need. I mean, you can only do so much in a semester-long course in professional skills, so it will require continuing opportunities to refine and develop those professional skills in terms of workshops, in terms of internships, the whole shebang, so to speak. More in the technical field, it's a great question. We cannot keep adding new things to learn without taking out things we do not need to learn anymore. And so I'm teaching a course this semester on breeding for quantitative traits in plants, and it has prompted some serious thinking. In the next edition of this textbook, maybe the last edition of this textbook, for example, I've proposed that we do away with the concept of heritability. Do we even talk about heritability, or talk about something else? You know, one change is mating designs. In the new textbook edition, I no longer talk about a diallele or a design to factorial mating design for estimating genetic variances, because nobody does that anymore, and there are other ways to do it. And so again, we cannot just keep adding things without taking things out in the curriculum. And it's a tough question. It's easy to come up with what do we add, but it's so hard to say, oh, what do we take out? Because, oh, I'm so attached to this topic, but you know my drift. And it's this whole topic of change, and we need to adapt to the changes that we see around us.
Well, thank you, Rex. That was an amazing hour. Now that we're coming to the end of the hour, but I'd like to thank you for all of us for attending. So.
Well, thank you so much. Again, I appreciate the time I've shared with you. Thanks to everybody who tuned in. Sorry I wasn't able to go. So I see there's a chat and there's a Q&A. Would it be possible, Lance, to save the contents of the chat and the Q&A so perhaps I could reach back out and try to respond to those that I didn't get to?
Yeah, I'm pretty sure I can do that. So I'll make sure I do that. So.
All right.
Okay.
Well, thank you. Bye now. Signing off.